think it's cool because you can actually. Well, it's an amino acid. If you really want to. Yeah, I'm scared. What's going on?
So if you need like a point.
I do, but it's only
Placebo.
No.
I'm still being
because I'm doing like
so it's not. Working.
I put a little sweet rat out there. I put a little sweet rat. Except, oh yeah,
We're
right
just like
there.
eating outside of
No,
Oh, my
that's
It's
no, it is, but the interesting thing is, apparently, these are the ones that we were just talking about this week. These are the ones that have all of the critical travel. So like CRISPR is not good for this, even if it would be more accurate. You, that is another case. So promising. Yeah, actually, we were saying that we're so
Wait.
happy that we're doing this because it actually makes us a lot more.
I'm so sorry.
Elizabeth and Clint.
I know a girl that's people survive. She's a snowballs at her. She killed. It's great. I was very good friends with this person. I'm not like a terrible. No, this girl was actually.
I teach it. Yeah, put it down here.
Why can't I? Oh.
So it's a meeting plus a.
Yeah.
No. It's a lot of. Yeah, you actually right.
Some states do.
Is that? You filming it? Because I have to leave.
Okay.
So, will I be able to view that?
Oh yeah. It's up right down if you want to watch it.
I mean, how?
Like you do.
I always feel like so many new things are happening. In China.
A little bit. The class that Robbie gave was classical. It's also advanced technology. Actually, I'm surprised with science.
Yes.
Yeah.
Fire heart.
They go.
I can get Ben. Ben? Ben who?
Ben.
Ben Griffin? With boots? What? In the Ben. The Ben. Yeah, dude, you're done. Yeah. He's trying to get me going. He sounded not interested.
Yeah, I didn't care. I knew you said it. I was like,
Yeah, why not? I'll learn something. Wait, Heim.
Are you going to be up here during that thing?
I thought I would start out and just like give a small podium, and because you guys were giving.
Heim, it didn't work.
Yeah.
Question.
Yeah, absolutely.
Yeah.
Because I'm wondering if you could take a picture, use my phone and take a picture. I mean, would that be okay?
Sure. While you're speaking?
What's that?
While you're speaking?
Yeah, while I'm speaking.
Yeah, I can manage to do that. That's no problem.
That'd be great. Look, here's my whatever. my password or whatever, because it'll lock up at some point. You know what I'm saying?
Okay. Yeah.
But that you know how to do it, right?
Yeah, I know it.
Yeah.
You know, right?
Yeah, I'm sure you
Connor,
know.
I don't think you need the password. You just swipe.
I know, it's, yeah. Thing. Hey, it's not working.
It's not. What do you mean it's not working?
It's a 1 second video of your face.
Click on it.
I just viewed it on the left.
Who wouldn't want a 1 second view of time more space?
Went to it, right?
You don't mind.
No. Videos, right?
I gave it a live one.
Oh.
Maybe it's a
Yeah, it's a little late.
Hey, notice you lose your voice like that.
Oh, I loved it.
I want to see how like that is.
I think I did. I totally did. Very late. This is after the I stayed till like 3 in the morning. There you go. I did it. Very nice.
That was the first one.
Yeah, jump there it is.
Yeah, you can have that back. I don't want that.
time it, so what all these comments, like, nice to be.
I'm really sitting nicer.
This is the thing.
Is this like you're supposed to be your thing when you do this?
So, I'm not setting this up. The
I
thing,
know, but you were like,
it was just
have a field trip.
basically, I was supposed to just bring people here. I didn't bring anyone. Yeah.
I think I'm actually gonna. And his lab partner is from like.
It's not a jail.
Like I was like actually.
Bare minimum. It's a reasonable estimate of like.
Hello, just to give everybody a heads up, I know it's past 6. We're just going to wait a couple more minutes in case anybody else is coming. Thank you.
Yeah, I was gonna say,
Cookie, anyone? Cookie? No? Come on, I know who you are. You want a cookie, I know you do. Yeah, you do. Yeah. You also want a cookie. Dude, I'm here for you, and only you.
Okay,
But you know what you do?
No problem. Cookie?
I did not do that. Okay.
Why do you have cookies? So basically,
Okay. Oh,
the
thank
thing
you.
my ISP uses our money for is cookies, so. Yes, we have Thanksgiving break and then
Hello, everyone. Welcome. Thank you for coming. I'm really happy to see this many people here interested in this event. My name is Chaim Moore. I am the president of the Biology Association. We are a student-run club here on campus, and we were founded with a lot of different goals. One of them being is to bring interest to biological issues, which is what we're doing here right now. And it's really important for us to engage the community as scientists. So we hope this is the first in a series of many panels to come. So if you enjoy this, please be on the lookout for more to come. I'm truly delighted to be here today as the moderator for this panel discussion on gene editing, which is a pretty big topic, so we have a lot of ground to cover. We will begin with a moderated discussion up here And then we definitely will have time to get to a question and answer section. So if you have any questions right now, or if any arise during the talks, then please save them, and we'll definitely have time to go to them. I think the goal today really is to focus on human health applications, but hopefully we do have time to consider some of the other applications of gene editing, because there are many. Some of the stuff I hope you leave here understanding is what gene editing really means. What is the genetic toolkit? What are the potential uses to combat various diseases? What are the impacts on society? And what are the ethical implications? So before we begin, let me just point out that no matter if you walked into this room strongly in favor or strongly against gene editing technology, or even if you have no idea what it means, you're in the right place. Today is not a debate, and the goal is definitely not to convince you either way. Instead, our objective is just to have a discussion on the scientific basis of the topic and go through different perspectives, the overview of the potential applications or concerns surrounding the topic. And for that reason, we assembled a panel with professors from many different academic perspectives to give us different insights on the topic, on the same one topic. So the four panelists that are joining us today include Dr. Elizabeth McClement, the one closest to me, a professor of molecular biology here in the Department of Biology at UMass Dartmouth. Her research interests include developing and advancing methodology to study the diversity in gene expression of microbial communities. I actually learned about gene editing from her in class, so I know for sure she's knowledgeable about this topic. Next to her, we have Dwight Duncan. He's a professor of legal ethics at UMass Law School. He has a history of high-profile bioethics cases under his belt, including involvement in several U.S. Supreme Court cases concerning subjects such as partial birth abortions. Dr. Robert Drew is also joining us today, next to Dwight Duncan. He's a professor of genomics in the Department of Biology here at UMass Dartmouth. His research interests include utilizing genomic and quantitative genetic approaches to investigating adaptation in fish, so feel free to ask him about Finding Nemo, the movie. We also have Dr. Yale McGrath with us here. He's the Chancellor Professor of Sociology in the Department of Sociology and Anthropology, also here at UMass Dartmouth. His research interests include looking at the connection between society, the economy, culture, and education, with a specific emphasis and focus into the way science and technology influence society. So quickly, before I begin, I just want to say and ask you guys, did you know that gene editing trials have already begun in humans? Just this July, the first human embryos were edited in the United States, but we're late to the party. Researchers in China and other nations have been approved and conducted gene editing on humans for over a year now. Undeniably, the era of human gene editing has already begun. At this time, I would like to ask our biologist, Dr. Elizabeth McClement, to come up and maybe give us a brief history of gene editing technology and how it works. Oh, sorry, quick thing. We are recording this and live streaming. The link is in your program if you haven't gotten one, they're up there. If you want to join the conversation online, you can invite your friends. Thank you.
Alright guys, sorry, but you have a little bit of a lecture first. Some of you guys are probably sick of hearing me lecture already. I promise I'm going to make it quick. So here's the basics of gene editing. So far, this is what we can do. First, we can identify places inside the human genome where we want to make changes. Now those changes might be taking a bad gene or a gene that has a problem with it, or a gene that's causing us trouble out, and closing up our DNA again. Or it could be that we want to cut into our DNA at a very specific site and insert a gene of interest in there. Perhaps that gene is something that replaces a non-functional gene that we have, or perhaps the gene of interest is something that helps us do something that our body is unable to do. But in any case, all of the gene editing technology has to cut into our DNA, open it up, and then do something with it afterwards. Now, the two different tracks that we have are cutting our DNA in a specific place and disrupting a gene that's giving us problems. So let's think about this in terms of HIV. If you have HIV, HIV has entered your white blood cells and it's stitched itself into your DNA. And when that happens, you always have HIV in your DNA, even if you're not actively producing the virus. So if you're on antiretroviral drugs, you're not producing the virus, but you still have it inside of you. And let's say you want to disrupt HIV completely. What you can do is find it in your genome, cut into it, have your DNA sort of break open inside the virus, and then when your DNA knits itself back together, it might do it imperfectly so that the virus no longer works. So disabling something would be a really good example of that technology. Where it could be that you would like to have a gene that helps you do something that you're currently unable to do. And in that case, you might want to cut your DNA open, spread it out, have a new gene inserted into it, stitch it back together, and then you have that gene forever. So every time your cells make a copy of themselves, that gene is also copied. So that gene becomes part of whatever cell line you've inserted it into. Okay. So we've kind of been messing around with gene editing since the 1990s. And there are these two things called zinc finger nucleases and talons, which both kind of make me think of like cans. And I think they're really cool names for them. And in both of these cases, what they do is they're genetically engineered proteins. We kind of made them up in the lab. And what you do is you sort of deploy them, sometimes in a virus, and they get sent into your body. They find a specific stretch of DNA. They identify it. They stick onto it, and then they cut your DNA open there. So basically what you're doing is you're sticking in a couple of man-made proteins with a target DNA sequence, and then something that can cut your DNA. And these things have been used fairly successfully in test tube studies. So if you take some cells out of your body and you want to edit them, this works pretty good in a test tube environment or in vitro. In some cases, you can just leave the DNA and let it repair itself. In other cases, you can send in a new gene and have that new gene be inserted and then use it for yourself. And by the way, you guys, just today, November 15th, this was all over the news. For the first time in California, we've actually used the zinc finger technology to repair, or hopefully repair, a gene inside a man who's sick with something called Hunter syndrome. Now in Hunter syndrome, he has a gene that is sort of disabled, and he's unable to break down a certain type of carbohydrate. If that gene could be fixed, he would be able to digest that carbohydrate, and his body would not be, his body would actually heal. And he's been struggling. I think he said that he's been through surgery every other year of his life. So he's constantly in the hospital, and he's constantly ill. And the illnesses that he faces are pretty severe. So when they treated him, what they did is they put a perfect copy of the gene so his gene is sort of his gene is ineffective. So they put a good copy of the gene into a zinc finger system, right? So the good copy of the gene is getting sent in. And then they're going to identify the place inside his own genome where the bad copy of the gene is, cut it out, put in the good copy, and see if it works. What they did is they took a virus, and they put this man-made zinc finger system inside a virus, infected the man with a virus, and these viruses headed straight for his liver. They're only going to infect his liver cells, and once they get in there, they're going to deploy this man-made protein. That protein is going to identify the place on his DNA where the defective gene is, cut it out, insert the right gene, and hopefully, if all goes well, he'll be able to start making the enzyme that he's lacking. So we won't know, apparently, for a month if it's working, but he was just infused with the viruses on Monday, and they just announced the news today. So this was front row headlines, November 15th, 2017. So that's pretty cool. All right, the problem is that these are not incredibly specific for the kind of DNA that we're looking for. So for instance, if I infected you with a virus, and that virus was trying to hit a sequence of your DNA, it might hit the right place, and you'd be great, or it might hit the wrong place, and you might disrupt the middle of a gene that you actually need to live your life. So if these genes hit in the wrong place, then you could run into problems such as cancer, disruption of a gene that you really need to do something, or perhaps it just wouldn't work when you've gone through a costly, really expensive therapy, for no reason. So one thing that we really need to do with gene editing is to make our targets really easy to hit. And the way we do that, nowadays is through the CRISPR system. And I guess most of you guys have heard of CRISPR. If you've taken a class with me, you certainly have. So the CRISPR-Cas9 system is something that we stole from a bacteria. Bacteria use this as their adaptive immune system. And what bacteria do is when they get infected with a virus, that virus injects them with DNA, and the bacteria takes a little piece of that DNA and it sticks it into a place in its own genome, in the bacteria's genome, called CRISPR. So they have all of these different snapshots of all of these viruses that have infected them in the past. If they face that virus again and get infected, then they whip out the little piece of the virus that they had saved from last time, they make a copy of it in RNA, and then they deploy that copy of it in RNA on a protein that can go and seek out that virus, grab onto it, and cut it into a million pieces and disable it, hopefully. So since bacteria have been doing this for ages, we decided that we were going to take this bacterial immune system and try to use it for ourselves. And actually, it's really, really well targeted. In other words, when we give it a piece of DNA and we tell it, go find this DNA and destroy it, we can inject it. It's okay. We can inject it into our body, and it is really, really good at identifying the target inside where we want it to hit. Much better than zinc fingers and talons. So the new CRISPR system is something that bacteria have already been doing. It's natural rather than man-made, except that we are giving it the target of interest. So for instance, let's say that we want to take HIV out of somebody's genome. We can, by the way, the white part is the is the Cas9 protein, which is the thing that's going to cut the DNA. The blue part is the DNA, and the red part is the little copy that's being carried around trying to identify the gene that we're looking for. So in this case, this red part would be a copy of the HIV virus, and we would basically be deploying this into our cells, and go and find the HIV virus, and when you find it, cut it out. Now we've tried to do this already in test tubes with white blood cells taken from people who were HIV positive, and it worked. But the problem with trying to get a virus out of your genome is that a virus is capable of mutation, and the HIV virus mutated exactly at the point where the CRISPR-Cas9 system was trying to cut it. So far, this hasn't been super effective in removing a virus, but it could be really effective in removing other things, like a gene that we simply wanted to remove. So people with Huntington's disease, for instance, have a stretch of DNA that's repeated too many times. If we wanted to just identify that stretch of DNA, cut it out, and stitch the DNA back together, that would be a really good target for the CRISPR system, for the CRISPR-Cas9 system. So the thing that we saw today, oh, by the way, I also found this. I hope you guys don't mind me showing this. I saw this. This is some of the first microscopic images of the CRISPR-Cas9 system in action. So the Cas9 is the protein that's going to be doing the cutting, and that little big pixelated yellow line is the DNA. And what this microscopic image is showing is the protein grabbing onto the DNA, having the piece of RNA that matches up with it, and then eventually cutting and throwing out the target DNA. The reason that CRISPR is one of the more, I don't know, I guess like modern and probably going to be one of the most upcoming effective ones is that it's a lot more targeted. So instead of accidentally hitting the wrong target, you could actually deploy CRISPR into the human cell and in theory disable or change multiple genes at once. So if you think of somebody who has a whole bunch of genes that are going wrong and not just one, you could actually deploy this and do gene therapy or gene editing on them, you know, on a pretty massive scale. It's also going to be cheaper in the long run to produce, and because it's already sort of been bench tested within the bacteria, you know, immune system itself, we already know that it works. Whereas the zinc fingers and the talon were completely man-made, like we kind of created them ourselves. But I was really encouraged to see this in the news today because it is, well, it's actually the first time that humans have been injected with a virus that had the CRISPR system in deliberately to change one of the genes around, and I'm really excited to see what happens to it. So I know that Dr. Drew is going to speak more about the applications, but one thing I do want to say is that some of the dangers of this are that it's never been tested before, and that there's a lot that can go wrong when you are changing your genes. If you're infecting an adult human being with a virus that's going to change some of their genes, then the only things that will be affected by that change are any of the cells that they reproduce. But there's also the possibility of doing this in an embryo. So so that when the embryo becomes a baby, all of the cells in its body will be genetically modified, and that comes with a completely different range of thoughts. It will probably be more effective as therapy in embryos, but that also brings up a whole list of ethical implications that go along with it, which I'm sure other people will speak to. So that's our technology where we stand right now. We have 3 really good systems. I think one of the reasons that this is one of the older systems is because it already made it through FDA approval for use on humans. So probably about 10 years from now, we're going to see the first thrust of the CRISPR system being used in humans if in fact that gets FDA approved. Now in other countries, they're supposedly already doing it. So like China has already said that they've been using CRISPR to edit embryos. I'm not really sure, and we haven't seen a whole lot of results come out of that yet. But I'm really excited to see what happens down the line.
Yeah, I'd like to invite Dr. Drew to come talk to us more about the applications.
Hello, everyone. Thanks, Simon, and the Biology Association for putting this together. Elizabeth covered the biology of it really well. I wanted to kind of cover some of the recent things that have been going on, but also talk about, actually I got pulled into the ethics quite a bit more than I thought I would, or maybe that's not the right way to phrase things, but I was more excited about reading about the ethics than I was the science. That's what I really want to say. So this is a really exciting technique. It's got this high specificity, and it is very easy to tailor this system to whatever gene you want it to do. So with the zinc finger proteins, or zinc finger nucleases, and the talons, each protein, that protein has to be tailored, or engineered so that it's going to recognize a specific DNA sequence. And that's not so easy to do because you have to get the right amino acids together in order to get it to identify that specific sequence. With the CRISPR, you just have to design this RNA molecule to match up with the DNA. So and then the Cas9, the actual enzyme that does the cutting, can just go where that guide RNA is, identify that sequence, and then make that cut. So it's a really effective system. It's the number of studies just doing research in the lab have exploded, and it's hard to keep track. Preparing for this meeting, it was overwhelming, the number of studies and ways that you could actually learn more about CRISPR. It's just overwhelming. It's also, of course, got some amazing promise for treating genetic diseases and cancers. It has not, as Elizabeth said, it has not been really deployed in these yet, but just being able to target those specific sequences and make changes, either cutting out the gene or putting something brand new in, is really exciting. And next, let's see. Right. Now, I feel like there's a lot of hype about CRISPR. It's for the specificity, it's really amazing, and a lot of people have gotten involved in this and excited about this. You see a lot of these studies. CRISPR pops up in the news quite a bit, and I think this is very similar to the hype that gene therapy got back in the 90s. So back in the 90s, the idea of going in and almost doing surgery on the genome, so fixing a problem in there, was like one of these great things that we were going to be able to do, and it was going to fix all genetic diseases. We were going to have to worry about genetic sickness anymore. Unfortunately, well, no. So the first human trials actually started in 1989. First human trial was in 1989. And we did not have an FDA-approved therapy until when? This past August. This is the first FDA approval for the clinical use of gene therapy, not CRISPR, but gene therapy to fight leukemia. So it's been almost 30 years since clinical trials started before we actually have an approved technique. So what I'm trying to say there is that we have had all this excitement about gene therapy, but it's actually a lot harder to implement than it would seem. You have to be able to get you can modify DNA, but you have to be able to get all the machinery into the cell to do that. And so one of the ways is by using a virus. So this is an example, an image showing using an adenovirus, which is similar to the cold virus. So basically, the virus would contain the proteins that are doing the modifications, that are doing the cuts, either finger proteins or talons or the CRISPR-Cas9. It would also have to have that guide RNA, and it would have to have in a lot of cases, you're going to replace that piece of DNA, so you'd have to have a DNA copy of a good DNA copy of the gene. And then you would infect either cells, maybe a cell culture, so that you can put those cells into the human, or you would actually infect the human with these. So the one that has been approved, which is the fighting leukemia, basically what they've done is they've taken T cells out of the human, or the patient, the actual patient who has leukemia, cultured those cells, and then used a virus called the lentivirus, which is a retrovirus related to HIV. But they've engineered that virus so that it can then deliver genetic code, they can change the codes, and now those T cells will actually attack cancer cells instead of what these T cells would normally attack. So really incredibly powerful. Basically, you're making the patient sick because they show all the symptoms of a sickness. They have fever. It's basically their body is acting as if it's sick, but it's attacking the tumor cells. Very, very cool. Very exciting. So it has been used in some leukemias, some lymphomas, as well as some melanomas. It's been pretty effective there. I've read that it's less effective against solid tumors, but they might be able to play with that, get those work. So this is not necessarily a magic bullet for curing cancer, right? It's worked in some cancers. You're going to have to try it on others. The other gene therapy that is close to being approved is for childhood blindness, a rare form of childhood blindness. This is a technique that uses, basically the gene therapy is packaged in a virus called an adeno-associated virus. So it's a naturally occurring human virus. It actually infects some primates, but it doesn't seem to cause any disease. And the virus will insert in a specific location of the genome on chromosome 19. For some reason, there's an insertion site there. So basically, you can package, what they do with this childhood blindness is they package all the machinery necessary to do the gene therapy. So they've got a good copy of the gene that would restore sight, stick that in the virus, and then they inject the virus into the eye. That's probably the least fun part of this. Under, I think it's general anesthesia, they inject that virus and then it infects the cells in the eye, delivers a good copy of the defective gene. It's going to put it somewhere else in the genome, so we're actually creating transgenic cells. We're not gene editing, which would be changing the existing gene. We're introducing a new copy of the gene somewhere else in the genome. And that has actually been very effective at restoring partial sight. So they're not getting our level of vision, but they don't necessarily have to walk around with a cane or have assistants walking around. So it's actually been effective for restoring, at least partial restoring sight. So very, very cool. There are some gene therapy in the beginning actually had quite a few setbacks. So the first virus one of the first viruses that they tried using, basically they were using it as an insertion tool. So instead of putting in proteins, you can use the virus, because some viruses will naturally insert into our genomes, like the adenosine-associated virus. So they tried using retroviruses to do this. And when they did that, they tried using this for developing a cure for leukemia. No, for SIDS, which is severe. No, not that SID. It's SID. Severe combined immune deficiency. Yes. So they were trying to correct the gene for that, maybe just insert the gene somewhere else in the genome. But unfortunately what happened is the virus they used actually inserted in random locations, and in some patients would insert near a gene and then triggered leukemia. So some of the patients, 2 of the patients actually developed leukemia. So using that virus was not a good choice, and this was actually this is of course very tragic for those patients. They were able to treat them for the leukemia, but you don't want a cure that has the potential of causing cancer. Another is that sometimes these viruses will actually cause, I mentioned they cause sickness type symptoms. There was actually a death in 1999 from a patient who was given, with using the adenovirus, that he developed severe reaction to the adenovirus and actually died from those symptoms. So he died from the virus, not from the gene therapy. So he died from, or the septic shock caused by the virus. So that's one of the things that has me concerned about moving too quickly with CRISPR. I'm very excited about it, but I also feel like if we put too much expectations on this, we need to move carefully, make sure that it is cutting in precise locations, because sometimes CRISPR can cut in other locations. Maybe we're just not detecting those. And when those happen, they could cause severe problems, either cancer or something related to that. And there's very little data on whether just how safe CRISPR is going to be for doing these kinds of changes. I think I'll wait, because I'm assuming somebody's going to ask about designer babies. So we can talk about that, my opinions on that when that question comes up.
Dwight, given your background in bioethics, I was hoping you can comment on the ethical implications of all of this.
Thank you. I'm hoping I can too, actually. I'm learning things because I'm not a scientist, I'm a lawyer, and I'm sorry about the bad news. And worse than that, I'm a law professor, kind of. But I applaud hearing about today's development, the first instance of gene editing on a human patient in the U.S., and of course very interested to see how that turns out. I guess, my sense of it is, I'll start with a simple proposition, right, which is that not everything that can be done should be done. And I say it's a simple proposition. It's really, that's why we have laws. I mean, you may not like some laws and disagree, and we can argue about that, but I'm saying law has a place because of this, right, that not everything that can be done should be done. And ethics is similarly concerned with that issue, right? That is, ethics is reasoned reflection about good and evil, right and wrong, to basically indicate what should be done, right, or should not be done. And I guess I'd want to start with some rather general propositions, but I think they're important in a way, and hopefully they have some application. Right? First is a simple principle, right? To do good and to avoid evil. And I think one of the things that means is that we don't do evil things in order that good things might come from them, right? So you don't kill an innocent human being to save other people, I think, right? I mean, even if it might do so, because that's doing evil to achieve some good. And the second principle is sort of a related one. It's just the idea that the ends don't justify the means, right? So if the results don't necessarily vindicate the method that you used to get there. And finally, I would say, I mean, even though I'm a lawyer, I'm also a human being, and I think I'm living, last I checked. And so I guess I would argue for the Kantian principle that that human beings should be always approached with a certain reverence, meaning that you should never use them as mere means, but always respect them as ends in themselves, right? That's what it means to be a human being, right? I mean, to be entitled to that kind of respect. And, I was thinking, well, how can I illustrate this point? I guess, these days the media is preoccupied, rightly so, maybe with all the sexual abuse, sexual harassment, stories and scandals, right, affecting this or that other, usually public figure of some sort. And I think what bothers us is that, well, one of the things that bothers us about that, in addition to just being icky and kind of gross and outlandish, I guess, is that it doesn't respect, that this kind of behavior does not respect human dignity, right? Nor does it respect human equality, right? That it in effect is using people, it's manipulating people, using them as means to your own gratification or whatever, right? Your own perverted, sense of pleasure or amusement or what have you. I mean, but it's a violation of this principle of, human dignity and human equality. So I guess all that's kind of by way of a roundabout introduction to the subject of gene editing. And I guess I would want to make a distinction between therapeutic gene editing, like today's case illustrates, where the purpose is to repair, right, some defective gene or series of genes, and what maybe could be called enhancement, designer baby kind of gene editing, right? Where the goal is to increase the beauty, maybe, get the blonde, blue-eyed person, the Aryan, or the muscles, or the IQ somehow, kind of maximize all these things, and I think that project, it seems to me that project is quite distinct from the therapeutic one. And I'm totally in favor of the therapeutic one, I guess, but I'm saying I'm very skeptical of the other project. Partly because of the threat it raises to human equality, right? It's no accident that the leading kind of proponents of eugenics in the 20th century were the Nazis, right? But what people don't often remember is that the Nazis largely modeled their eugenics program on the United States, things that were happening in the United States, right? And, even very well-known progressives like Theodore Roosevelt or Oliver Wendell Holmes Jr., were basically social Darwinists, and so they believed in the survival of the fittest, and if you apply that to the political realm, I think it quickly leads to a kind of master race kind of thinking that the Nazis just took and ran with, right? So I guess what I'm saying is the designer baby project or whatever is a troubling one because it raises the specter of kind of master race ideology, and in effect the people who are disabled, who are ugly, who are whatever, that they will necessarily be viewed, be left behind and marginalized. And I think, well, I don't know, I just think that everyone is a human being and everyone is entitled to a certain respect because they're a human being, right? And in the eyes of the law and in the eyes of God, everyone is equal, right? So I guess that's my concern with the kind of enhancement use of, or prospective use of gene editing, right? I think the safety issues have already been alluded to, right? There are issues about of the safety of this procedure, and it seems to me those things have to be, have to be tested and kind of resolved maybe through animal, experiments on animals and so on before you start kind of monkeying with humans, so to speak. And also there's this, related issue is the issue of kind of the unintended side effects of this, right? That I think we have to have a good sense of what else can happen, right? In addition, while you're trying to fix this gene or whatever, that some other thing occurs that is horrifying, and this is particularly important when you're talking about affecting the germline, right? That is, that we're not just dealing with therapies for individuals, but essentially having human beings whose genetic makeup has been modified and may reproduce and thus pass on this modification, right? That's something that I think is kind of is a whole different scale of kind of concerns. And finally, I'll just allude to one of the problems which has to do with the means, right? The way in which knowledge, scientific knowledge is acquired in this area, right? One of the things the Nazis did, was did all these kinds of experiments on concentration camp prisoners, right? How cold does it have to be before they freeze to death, right? This kind of thing. And to a certain degree, they did gain a fair amount of knowledge of these subjects, right? But, shame on them, right? Because you can't treat human beings that way, right? And I guess, I think some of the work that's been done in this area has involved the creation and ultimate destruction of human embryos so that you can kind of monkey with them, right? And I'm just putting a big question mark behind that whole process, right? Because, in effect, it's saying certain human beings, admittedly embryonic, right, are totally expendable for the benefit of other human beings, namely you and me. And I just question that. Now, I admit that this goes on in plenty of other areas, right? The whole assisted reproduction business, right, involves the creation and destruction of human embryos, sort of surplus human embryos. So this is a bigger issue than kind of gene editing, but I'm saying I think a number of the experiments that have been done have involved the creation of, and destruction of human embryos, right, so that you can play with them in the interim. So I guess I'll leave it at that. Hopefully share some provocative thoughts.
So whether we're changing ourselves or changing others, this is going to impact how we interact with each other or look at some social issues. So Dr. McGrath, I was hoping you could comment, show us how this would impact society.
Hi, I'm Neil McGrath. I teach social impact of science and technology. I'm not a biologist, so I do not know exactly how well developed at the moment gene editing research is. Also, I believe within biology is debate, certainly it's debate within social science, of how much intelligence, skills, personality are determined, behavior are determined by genes, and how much is not. Can we define what are desirable traits? How do we encourage them? And the real problem is, what is a desirable trait? We can obviously talk about these things that Drew, right? Dwight mentioned. We can distinguish between therapeutic and enhancement. But the line is not always that clear-cut. What is desirable? Who decides what's disease? They're not that easy to define very often. We know, for example, of a serious disease, which probably does have an element because of children's chemistry. You give a child with ADHD, Ritalin, he'll behave differently. It does change biochemistry. But is that a serious disease? What it really is an 8-year-old who cannot sit still for 6 to 8 hours a day. Now, there is something seriously wrong with 8-year-olds who can kind of do that. There's nothing wrong with an institution that expects that. So the real question is, do we adjust people to society, or do we adjust society to people? And who's to decide? There was an unofficial, unplanned genetic experiment in China, the one-child policy. One thing we can clearly say is determined by genetics. Simply chromosomes is gender. Parents were restricted to have only one child. In China, traditionally, boys are more valuable than girls. Girl embryos were more likely to be aborted, and there probably was a substantial amount of female infanticide. Result: an imbalance of more men than women that, in fact, has produced a certain degree of social chaos by letting parents make decisions. Do you let the military make decisions? The military historically wants people who are aggressive, militaristic, and violent. And those are the traits you want to encourage. Do you let corporations decide? Corporations, like the military, want obedience and docility. Are these traits that you can genetically single out for? Maybe you can't. But if you can, as you kept asking, should you? Who's to decide? What does folk society need? What kind of power balance does it need? How much of each trait does it need? In the 1930s, Aldous Huxley wrote Brave New World. This is before DNA was very well understood. Embryos, children, were deliberately created to be alphas, betas, deltas, and epsilons. Alphas, of course, were in charge. Alphas were ruling class. Epsilons would do the least desirable work, and the others were in between. So the epsilons had to not question authority, had to obey, and not challenge, and they were deliberately genetically programmed to do that. Is this scientifically feasible now? Probably not. Will this be scientifically feasible in the future? Perhaps. But as you say, what can be done is not what should be done. The problem is, who is to decide? We live in capitalism. We live in military capitalism. We have military that needs people who obey orders and fight. In capitalism, we need workers who are docile, controllable, and don't form unions. So is that who we are going to be programmed for? And already I mentioned ADHD. We have a school system supposedly designed to educate children, but it refers to children who are docile and easily controlled. And not being docile, not being easily controlled, is actually labeled a disease. Now, what's desirable? We talk about beauty. Beauty is not an abstract thing. Beauty Beauty is a social construct defined in various social settings. Look at paintings. Probably up until the early 1900s, certainly going all the way back to the Renaissance, all the way back to the 1400s. The beautiful people were much heavier than today's, some people would say, almost anorexic models. Being, by today's standards, fat was called beautiful. And yes, fat does have a genetic component to it. That has use. I'm speaking in biology, not a biologist. At a time when there is a real possibility of malnutrition, when you may not simply get enough to eat, your body, a healthy body, will store more fat. So at that time, fat people were considered to be healthier. And go back to your grandparents, or perhaps great-grandparents' time. A woman is pregnant. What wish did she receive? Have a fat baby. Why do you want a fat baby? Well, once upon a time, maybe 70, 80 years ago, the most common causes of death were communicable diseases like tuberculosis, diphtheria, typhoid, smallpox. Some of you probably never even heard of these. If you get them, especially as a child, what you need is reserves. Heavier people have reserves. They are more likely to survive an infectious disease. What do people die from now? Because heart disease, high cholesterol, followed by cancer, both of which in your 60s, you are much more likely to get if you are heavy than if you are thin. But think back to a parent, say around 1900. Do you worry about your baby developing high blood pressure in his 60s? Or do you worry about him surviving measles before he's 5? It's not a concern. Now, because we live in a very different world, and this very different world has a very different concept of what is desirable and adaptive or not adaptive. There are other diseases, really dead diseases. Sickle cell anemia. It turns out sickle cell is genetically helpful in the event of malaria. You live in a malaria-infested environment, you get sickle cell, you are more likely to survive it. So again, evolution is going to select people who might live to 20, might live to reproductive age, and then die very soon afterwards from sickle cell. It is not going to select people who are going to live in their 80s and 90s, because no one does. Malaria and infectious disease wipe out first. But these are real possibilities for us. So we have to figure out what really is therapeutic and what is enhancement. The line is not at all clear. And who's to decide what is enhancement? We can talk about a behavioral disease. one I think will agree is more serious than ADHD. And I'm amazed that ADHD is called a disease. Willingness to challenge authority, willingness to be creative, willingness to think for yourself, having a lot of energy, that's a disease. But there is autism. Now, autism, real autism, I emphasize, real autism, you have no contact with the outside world. You normally cannot develop language. You normally can never be left in a room by yourself. You have virtually no awareness of your environment. You have no awareness of who you are, maybe even no awareness of self. They say autism is on the rise. Well, it is because autism has been redefined. And there is a new category of so-called mild autism, mainly Asperger. And frankly, as scientists, that a good percentage of you have Asperger. Terrible disease again, just like ADHD. A kid who prefers playing with Legos to sports. It has some vague similarity to autism, and yes, it is part of an autism spectrum. But is it a problem? Why is that called a disease? Why is that something that should be controlled? So it is really social that determines what's called desirable rather than what really means a better or worse life. And who gets to decide how we make that policy, how we're ready to walk, how are we to build schools where ADHD and Asperger are not seen as troubling and diseases. Anyway, should I stop now? Okay.
Thank you to all the speeches from our panelists. I'd like to open up the time now to our panelists. If you guys maybe wanted to address each other, you guys can talk freely. Yeah. It should be. Yeah.
All right, I'll start. I thought these talks were really eye-opening, and two things that particularly struck me is one, I don't know how you guys feel about this, but if you change the genetics of a developing embryo, you're changing essentially the human genome, right? Because that embryo will turn into a baby, will turn into a reproductive age adult, and when they reproduce, they will pass that genetic change on to their offspring. And perhaps changing a germline gene will change the future of human evolution. And one thing that Yale said that really struck me is sickle cell anemia is a very useful gene to have in our genetic arsenal. But it also causes a disease, so it might be the sort of thing that we would have targeted for removal without maybe realizing that at some point we could have used it for something incredibly useful. So I think it's a really good question to ask how we are going to make up our minds about what genes to remove when we are so ignorant about how those genes sort of play into the whole organism. In some cases it's cut and dry. If somebody's dying of cancer, you want to save their lives, okay. There are some parts that are really cut and dry. But once we get through that cut and dry area, we're going to very quickly enter a gray area. And I think that's that's something that personally makes me nervous, and I don't have any answers to it. So I just kind of wanted to put that on the table as something that I'm completely, I don't know, I'm sort of morally confused about myself.
As far as I know, malaria is likely to be cured. So it probably is not something to worry about, so you probably want to avoid sickle cell, but of course, what do you do with sickle cell? As of now, if you hear you're making a sickle cell, that means they don't have the opportunity to be born, let alone grow up. This is something that warrants abortion. But I think the point you're getting at a little bit, and I really understand, we really don't know what exactly. We do know that evolution selects certain traits that many of us don't understand. For example, the appendix. People for a very long, biologists, everybody else are not. Biologists for a very long time did not know what the appendix does. Did the same with tonsils. They removed it. It was a big mistake. Things may be there for a reason that we don't know about. But also, again, the problem, I mean, I can think about things like, for example, the famous case is Angelina Jolie. She carried a gene that made herself vulnerable to certain types of cancer. So she did a complete hysterectomy, she moved her breast, on the possibility that she could develop a cancer at some point she not yet have. Sometimes genetic knowledge may end up doing more harm than good.
I guess another thing to think about is we have a complete human genome sequence. We've got an estimated 20,000 genes. We're the product of all of those genes interacting with one another and interacting with the environment. And predicting how, if we do a change to those genes, how that's going to actually affect the organism, whether we're doing this at the embryo level or even at the tissue level. That's actually quite daunting, especially since we don't know what we still, let's see, it's we're about 10 years since the human genome's been completed. Human sequence, we still don't know what half of those genes do, what most of the genome does. We don't know what it does. And making changes, especially at the embryo level, for genes that we really don't know what's going on, we can't really predict what's going to happen, especially if we add evolution to this. And actually, that's something that I ran across quite a bit when I was Googling this, is that there are lots of articles about scientists wanting to put a halt or moratorium on work on embryos. So even the people who invented this technique are worried about the kinds of directions that it can be taken. So and I think that's actually really positive that we have discussions like we're having today, where we're questioning whether it should be done, because that means, hopefully, that we're going to make very careful decisions about how it is done. That's really what's important. I'm almost less worried about what happens to humans, human gene editing, than I am about, it's almost like a Wild West when it comes to other organisms. So we can modify other organisms. There's a little bit, everyone's afraid of GMOs, so that's when we introduce a gene that's not in that organism to begin with. So like antifreeze protein into strawberries, and then they don't taste like fish, but they don't suffer from freezing, but they're now something different, and everyone's worried about that. We can think of humans who undergo gene therapy as being GMOs. And honestly, there is no evidence that GMOs are actually bad. But when it comes to things like the CRISPR, so there have actually been crops that have been modified using CRISPR, like button mushrooms that don't brown. By knocking out just a single gene, they decrease how they brown, so they actually stay fresh longer. It's turned out that the USDA has decided that they don't have to regulate that because there's no new gene put into it. It's that it's genetically modified. They've knocked out a gene, but it's not regulated because it's not a GMO. It is if you use the strict definition, but there is actually a legal definition that it's something, an alien DNA put into that organism. And they're talking about doing things like controlling mosquito populations to fight malaria or Zika, by releasing genetically modified mosquitoes to the environment to decrease the population. But can we predict what's going to happen to that ecosystem if we So and these kits are actually, you can almost buy them on Amazon.
You can buy them on Amazon.
Yeah.
Did you know that you guys can buy a CRISPR kit, a CRISPR gene editing kit on Amazon? It's $137 when I looked yesterday, but they don't ship fine.
That means a grad student or an undergrad in their garage could make something brand new. And we don't have any laws right now that are governing this. The USDA doesn't feel like they do, but the FDA might feel like they can regulate it. But right now, the USDA does not feel like they have any authority.
Right. I'm sure many of you have questions. I think we're a small enough group that you can just raise your hand and speak out loud. So let's get this started.
Alright, so in biology we know that DNA polymerase, I guess this is for we know that DNA polymerase has percent success about 99%, and that 1%, 0.1% thing, that causes mutation and overall causes lead to cancer. So I know, so a biochem major, I know that biological molecules sometimes don't work because of the environment, pH, temperature, anything. So CRISPR, what is the percent? Is there any percent error? And if there is any percent error, are we, I guess, like, do we really want to introduce that error to our body?
Right. That's an excellent question, and I think that's where there still has to be more work done. Even just doing the tissue-specific therapies is just understanding how specific is this. So I think it's at least right now the evidence is that it's much better than the other types of techniques. So relying on the machinery of a virus to insert randomly in the genome, that's more prone to error than CRISPR right now, at least in terms of modifying cells or modifying research organisms. But I don't know if they've done enough experiments to actually measure that error rate, because if even a small amount of error is going to impact that individual.
I'll also say that the CRISPR system isn't meant to polymerize anything. You're not making anything. You're basically disrupting it and then sticking in something else. And in some cases, you actually want to disrupt it. So you want to cut the DNA, and you don't want it to repair perfectly, because if it repaired perfectly, you'd be back at the starting point. You want to disrupt it a little bit. So the enzyme itself is just cutting. It's not trying to make any new DNA. It's basically up to us to give the new gene that will be, effective in getting added to our DNA. And then it's up to our polymerase to make copies of it from that point forward. So it'll be subject to the same error rates we're thinking as any of our.
So I just want to address some interesting comments that were made about the panel when they speeches. I guess I have more of a personal investment in this because I myself have a genetic disease along the lead attack chromosome called Hirschsprung. Forced like about a foot and 3 inches in my intestinal tract to not have like the nerves that regulate it form. Basically caused a lot of issues, having a lot of surgeries done, etc. So knowing like the way I've lived with that, my own complications and I think very much of human dignity. And while mine is like relatively non-severe, obviously I'm still living today and I'm fine now. I've had surgery done to rectify it. It's still something I would be like, yes, if I had the chance to genetically edit it out and get rid of whatever that complication was, sign me up, you know what I mean? I would take that option because knowing the struggles I've had to go through with it. And there have been some, like granted it's still survivable, but it hasn't been exactly comfortable and it has robbed me of some of my human dignity in some sense. Some robs do exist today. So when I think of that, I also think of like maybe it would include something. It would be important for us to put it to each individual. And I understand there's been a large body of text in literature and the societal reaction to this. But there was also that to humans. Excuse me, to computers. When the first computer came out, everyone was terrified. Big black box that took up a whole room, 8 word, very much in a very reactionist stage where we don't understand a lot of what's going on. And I think that it would not do like over to jump the gun basically. But to put my question forward, to ask about the article that was treating Hunter syndrome, I believe, they mentioned that the man was having like on and off surgeries every other year. So if there were attempts to like rectify the syndrome problems, speaking from my own example, if you then try to introduce genes that can rectify my disease now, I'm missing that section of the colon. So trying to reform nerves would then like have the added sub effect, like trying to reform nerves along the whole colon, along the whole intestinal tract as well, even though I'm missing a substantial part of it. You would have to code in for something like that as well, knowing that here's a past list of surgeries, here's a past list of what we go through. Will the genes still attempt to code for this, even though some things are missing, some things are there?
Right. So that's definitely true that gene therapy is going to work on for some diseases and not for others. So and it may even have limited effects on those diseases. So in an existing adult, you can really, it's in order to really something like what you had to be fixed while you were developing, rather than now. It may not be something that gene therapy could actually repair now. It might be something that, as a person, you might consider gene modification. I'm throwing it out there, not saying I support it, but gene modification so that you don't pass it to your offspring is one other thing to think about, that you would pass healthy alleles or healthy genes to your offspring. But that's definitely true that gene therapy is not going to cure all illness. That even some genetic diseases can't be repaired once we reach adulthood. It might require changing way too many cells or already passing that time point where the repair can actually begin.
I think what you said is really interesting, though, about every individual, and I'm wondering how you feel about this, that every individual should have their choice whether they want to change something rather than having a government mandate that says you can or can't. Gene therapy can fix your problem, and somebody told you, no, you can't do that because we don't want to, create a master race or say that you're not perfect the way that you are. What purpose of personal preference?
The problem is, very often the choice is not made by the individual, especially in embryos. It's made by somebody else.
That's true.
So you have to figure out who has the authority and what is desirable. And as you say, Elizabeth, there are certain traits that have both a desirable and undesirable side to them. And how do you weigh it? I mean, and frankly, I think it's a very good chance if there were the school system had complete freedom, it would eliminate ADHD, and it would eliminate Asperger's, and the process probably eliminate both creativity, most innovation, and most independent free thinking. And we're talking about the American school system. I think we'll do that good.
To respond to that, like I understand Asperger's and ADHD both are diseases that have some negative effects as well. This is why we're more out of type with this argument, because they do demonstrate negative behaviors as per the DSM-5 that do interfere with normal functioning. It's why it's not all like you're just an outlet of creativity or an overall burst of energy. They're a general disregard for normal logical functions that are necessary to survive and function on one's own, and that's why they're classified as disease. But going to like the idea of like, well, who's calling the shots on this? I bring up the idea of like smallpox that you mentioned earlier. That was a forced mandatory vaccine.
Yes, it was.
Right. So the idea was everyone knew this was negative, regardless of your own personal beliefs about getting a shot, even if you were afraid of needles or just didn't want to take a vaccine. Or, for example, later on, if we had like people at the same time believe maybe autism is caused by vaccines, it was still mandatory because everyone knew that this disease was so deadly that we had to get rid of it. I think this falls more into that category than dealing with ADHD and ADD.
Autism is a good example. First of all, I think I need to point out, I think you did, a lot of parents are very resistant to having their child vaccinated for things like measles, German measles, mumps, for possible reasons. It's a real distrust medicine for a reason. But I think autism is a really good example because I have direct knowledge of one case of what with autism, and it is very hard to deny that is a problem that if it could be cured, it should be. And I suspect a parent who were told that your child would have autism, in the real sense of the word, may very well choose an abortion. But we no longer know just have autism, we now have autism spectrum. Sometimes mild autism, quote unquote, is given a label to be called a disease. You're on the autism spectrum, you have Asperger, and that means that you are overly focused, you might choose Legos over sports, you like to be in small groups rather than big crowds, you like to have a small group of intimate friends rather than many associates. You may like to read, you may like to solve puzzles, you may like to do math. That in fact may have some genetic link to autism. It may. But Asperger's is just a personality trait that has many very desirable characteristics. Now, I'm not sure if there would be genetic tests that could distinguish real autism from Asperger. I guarantee if they chose that way, I'll bet Einstein would have been aborted.
Actually, this brings another level to this about what kinds of diseases can actually be treated with gene therapy. So even if we consider autism to be a disease that can be treated by gene therapy, autism is actually a disease that's the result of multiple genes. It's not just a single gene. So when we're talking about sickle cell, we're talking about a single gene, we go in and fix it, no more sickle cell. But with autism, or if we're even talking about designer baby type traits like IQ or height, things like that, that's controlled by hundreds of genes with very small effects on the actual trait. And that's where we get, and we also have those interacting with the environment. So smart people don't necessarily have smart kids. It's that kind of thing. There are environmental effects. The same with autism and that whole spectrum is it's the result of different combinations of alleles that may be, or variants of the genes that may have detrimental effects on behavior when they're all there, but a few of them have good effects, and they may even be having a selective advantage. That you do better in school, so you get a better job. Wow, there may be some advantage to having some of those, what we would call disease alleles. So eradicating all of them could be bad, but having all of them together could be bad.
And when do you know? How do you know which time you cross the line?
And even, but if we brought gene therapy into that, changing one gene that has one little effect is not going to be a big change either. So in some ways, even having the discussion about designer babies, if you're doing a really simple trait like eye color, maybe, but if you're trying to affect IQ, it might not even be possible unless you want to spend money to do hundreds of genes and change all of them to what you think is going to be.
And I'm pretty certain Asperger correlates positively with IQ.
But that's a good point. That is one of the powerful ways we can use CRISPR and gene editing is to experiment, do basic research, and figure out what is the cause of these genetic diseases.
In model organisms.
Right. So just because they can't act as a treatment now, because we can't fully understand some diseases, by using it, by utilizing gene editing, we can find out more about the disease and eventually hope to treat it in some way.
I know Dr. Drew, you brought up the fact that I think leukemia was just approved to be for gene therapy.
Yeah, August 3rd.
Oh yeah. So like, it taking 30 years. Does that mean it's just for human trials, or is it like commercial?
I would say it's a series of setbacks. So as of 2012, so it's very hard to find out how many trials are going on. Things like that. There have actually been, so as of 2012, so that's 5 years ago already. There were 1,800 clinical trials going on across the world, and not all of those are going after the same disease. And some of them are going to have setbacks. So the clinical trials may not work as effectively as it did in mice. Those kinds of things. Because we find that actually they're finding a lot now that things that work in mice don't necessarily, and often do not work in humans. So I think it's more of getting through those hurdles to finally getting to an approved protocol. And actually, there are two that are approved in Europe. One is for the severe combined immune deficiency, and the other one, I think, is for the blindness. I think the blindness treatment is going to be soon approved here. But it's hitting those setbacks, and there are a series of clinical trials that have to be performed for any drug or treatment like this. And they have to make it through those hurdles well. They have to do more harm than good, because there's risk associated with that. So this treatment should work effectively. But it's just taken longer to actually get to that point. I think, though, that CRISPR might accelerate things, where we can actually make those targeted changes, the therapeutic targeted changes, more quickly.
So I was thinking from the 90s till like now, it's kind of a long time. 20 years. If we're, or 20 or 30 years, if we're, I feel like maybe the FDA might kind of be holding us back. But if you're saying that, we have to show like good results consistently, I guess
we're altering the DNA, so that's, like Elizabeth said, that can be, if it causes cancer, that's a very serious change.
I have a question. So we've been talking a lot about the ethics of using it on patients. One of the things I know is that people with terminal diseases, they have the right, I think in the US and most states, to use drugs that have only passed phase 1 through the FDA, in case it may save their life. I was wondering, maybe from like an ethical standpoint, you guys can talk about if there's really consent there, or informed consent, and if you think this should be allowed for CRISPR.
Yeah, I guess my sense of it is the law, in the legal system, in the whole medical area, the dominant rule is informed consent, right? So that patients can, as long as they know what the possible benefits, possible risks are, and they accept that, then it's okay, really. So I guess in terms of experimental drugs with people that are terminal, I guess I'm inclined to be quite liberal about that, right? It's not like they have much choice at that point. And so I guess I would think they should be allowed to use experimental drugs, assuming that they, are informed that they are experimental and that there is a downside risk that's associated with it. But I just think, for people that are, whose prognosis is that they're going to probably die within 6 months or a year, I'm not sure, how decisive the downside risks really are, actually, at that point. And I think they should be allowed to choose.
So to add to that, though, I think that there's another factor involved there that's not necessarily a legal choice about whether they can do it. There's also the economic choice. If we were just talking about gene therapy, a single treatment of gene therapy right now is estimated to cost about $500,000. Yeah, $500,000. And so that leukemia treatment, $500,000. So, or if you're getting treatment for blindness, it's $500,000 per eye, right? So very expensive treatment. And I think for someone who is terminally ill, I think then it becomes insurance will cover it. And will the insurance cover it if they're terminal? And it's experimental. So they may not actually have that choice.
I have one comment if I can add to that. Often I find patients who are terminal aren't allowed in the studies because they will ruin the outcome. So if you're terminal and there's a gene study underway, you're too sick to qualify for enrollment most of the time because they don't want negative outcomes on the study as one of the concerns. So we have a hard time getting patients into studies if they have become too sick to be able to be a good candidate for the gene therapy. But what you had said, sir, is what I think is one of the most important things. It speaks a little bit to what you were mentioning about disease processes, is that the Angelina Jolie who could have the double mastectomy
Yes.
and have her, she could afford to have it.
Yes, you
And
could.
I think one of the things we'll see is that as this moves forward, which I think for single gene issues it will be a big thing, is that the socioeconomic piece of it is very big too. It'll widen the gap between those who can afford to have editing and those who can't afford, so will maintain their disease status.
And of course, we are the only country in the world where medical policy is made by private profit-making insurance companies rather than a single-payer system that is designed for the good of the patients. So that's something to keep in mind. The other thing is that the more these practices become routine, the cheaper they'll be.
It's true, like when antiretrovirals first came on the market, they were prohibitively expensive for 99% of people who are HIV positive, and now, they're a lot more affordable, right? And we're no longer paying for all of that basic research that went into the development of them. Maybe gene therapy will be the same.
I'm actually a little concerned about that because a lot of these diseases are rare because they reduce our survival. So that means there's actually a small population that can actually benefit from gene therapy that's actually going to be paying for it. So how much is that going to affect the cost of the therapy?
And it'll get cheaper if we do things that are more cosmetic because more people will be in the market for them.
Right?
Yeah,
Yep.
rare diseases.
Like fixing tools or something like that.
Actually, I want to bring this up because we've all the idea of private insurance companies. One of the tricky things about getting insurance companies to cover your treatments and maintenance is to whether it's classified as a pre-existing condition.
Yes.
Or genomic mapping. That means a lot of that data is going to have to be stored somewhere. If you were to share that with your insurance company, you might be like, we'll see you have the markers for this, 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 and that. It's a pre-existing condition. We're not going to cover it. So we're going to have to change the laws about that as well. Because, and this would be a question for the law experts as well. Because if we allow that to stay as it is with pre-existing conditions being the case with denying coverage for insurance, saying that they don't have to pay it, a lot of these genetic diseases will automatically classify as that. Will we have to change current laws surrounding those?
Well, of course, I think Obamacare, right, required insurance companies to cover pre-existing conditions. Now, admittedly, the legal status of that is a bit unclear, or the future of that, but I think the trend has been definitely to require insurance companies to cover pre-existing conditions so that they would cover genetic issues, and I'd certainly be all in favor of that. I guess I'm a little bit, the question was rightly raised about who decides, right? And somehow, the specter of private insurance companies making these decisions, that's kind of considered something really problematic. But I would say the move towards government control is not necessarily geared towards patient choice, right? That one of it seems to me our medical system up to the present day has largely been a kind of capitalist system, so that if you could afford, or if you could afford the insurance policy, whatever, you got the coverage. If you couldn't, you didn't. Apart from Medicare and Medicaid and sort of certain exceptions to that, but clearly Obama made a move to expand that. But then of course, then instead of having insurance company executives decide, it becomes government officials deciding. And I'm not that confident, I guess, that the government is going to be that much more responsive, right, to individual needs and concerns than big business is actually. So I mean, maybe it will, but maybe it won't. And I guess I'm sufficiently skeptical of bureaucracies. You know, I know we're part of a state university system, and so we have a kind of big bureaucracy in our college life. You know, but I guess I'm sufficiently skeptical about bureaucracies that I'm not, you know, be it government or big business, I think what has to predominate is the concern for the patient's welfare, right? And really, generally speaking, the patient, him or herself, is in the best position to judge that, right? And in that sense, I'm in favor of sort of the move towards informed consent in our legal system with respect to healthcare decisions, right? That it's no longer just doctor knows best or, you know, insurance company knows best, but it's rather, you know, the patient in general knows best, right? I mean, hopefully the patient's informed.
We do have some legal protections. So there's the Genetic Information Non-Discrimination Act.
No, that's right.
Right?
Yeah, that's
So,
right. There is some, yes.
I can't remember if that applies to insurance companies. I know it applies to employers, but I can't remember if it applies to insurance companies. I think it does. That was 2008.
Yeah.
And some states have additional protections on top of that.
Right, we have time for a few more questions.
While I definitely find myself a lot more conflicted on the actual disease point, like the things that we want to get rid of, like things like sickle cell anemia, things like malaria, I'm more concerned with like the idea of designer babies. Now, if we invest into parents the authority to dictate what traits their child will like express as they go through their development, like their hair color, their eye color, things like that, as they're developing in utero, the world that child's going to kind of inherit is really going to be the one that their parents lived in for the most part. We'd expect the world to kind of change in 20, 30, 40 years. So the traits that those parents decide or some other entity decides are desirable or things that they want, those things might not be desirable anymore when that child is able to make that decision for their own child. So like I just want to understand kind of what the ethics, I don't know if that's a word, what that is. How ethical is it for a parent to be able to make that decision now, given that the technology will do exactly what they want when they want to do it?
A good example of that is, once upon a time, most parents would select for heaviness. Now they will select for thinness. And the pendulum may have gone too far in their direction. So how do we decide what is the desirable trait? And that's just a continued change. Just yesterday, they decided that anyone with blood pressure over 120 and 80 has high blood pressure. 3 days ago, that was considered the ideal.
So actually, does a child have legal, like if a child is born, do they have a legal, could they sue their parents?
In general, no. I mean, that is, in general, parents are assumed to take care of the best interests of their children. There might be certain situations, though, in which a parent has demonstrated, kind of unfitness for the role, in which a guardian might step in effect, to protect the child vis-a-vis the parents. You sometimes have this, for example, I don't know, with Jehovah's Witnesses who refuse to get blood transfusions for their children. The law is that they can make martyrs of themselves, but they don't have the right to make martyrs of their children, right? Or Christian Scientists have the same kind of tension, right? So, but normally, the children, are assumed to be, have their interests taken care of by their parents. So I think in the normal situation, children don't have.
In this case, you're making a choice for the children's children, and everyone that comes from their genetic line from that point forward.
This is what the troubling thing about the designer babies business, I think, is this, right? It's a sort of command and control model of parenting, and I'm just saying, I don't think that is actually what parenting is all about. I think parenting is welcoming a child and and educating that child for freedom, right? So it's really, and to the extent that you think you can program and control everything, I think that you're mistaking your role. In my view, I don't know. It just doesn't seem that's not an adequate understanding of what parenting is about, really. And I'm afraid this this business about sort of choosing traits that you want as opposed to traits that you don't want. I mean, it's one thing we're talking about virtue. It's another thing if we're talking about, I don't know, physical characteristics and so on.
And so to real quickly add to that, I'm actually very skeptical that designer babies will actually happen. But if you think about it, what was plastic surgery originally designed for? It was designed to help people who were in accidents to repair their faces. But what's it used for now? Where's the money-making part of it? It's vanity. And so I'm actually both skeptical of designer babies actually being a reality and worried that somebody's going to try it.
The man with the pencil.
So I guess my rebuttal to what just came up was like, is allowing random chance more ethical than making these decisions in a conscious way because we have the technology? Is making these decisions about hair color or eye color? Well, I also agree that I think the argument in general about designer babies is not very constructive to the relevant ethical questions here. But if a sperm bank only accepts sperm from male donors over 6'2", let's say, is that an unethical thing to have happen as well? Because we're kind of selecting among only a subsection of the population, or if we're only like selecting among physical characteristics of labs because we like chocolate labs instead of yellow labs. Like, because we have the technology to make these physical characteristic changes, in the end, I guess I don't find it as unethical as most people do, even though I don't think we'll actually do it in human beings.
I feel like those traits are the product of the long line of evolution in which they were selected for in our population, and so we are choosing, when we have babies with somebody, we're choosing traits that have already gone through the gauntlet of evolution. When you deliberately modify a baby, okay, maybe changing eye color isn't going to have a huge impact in the upcoming generations, but if we changed a gene that did have a huge impact, we're basically subjecting this line to a trait that perhaps hasn't been seen evolutionarily. Like I said, maybe not for a single trait like hair color or eye color, but if we started to try to change things like intelligence, and we are wielding a dull knife with it, then it's possible that we are introducing something into our children's future genetic line that is not, well, that will be subject to evolution in a pretty brutal way, right?
Would you question the use of CRISPR and gene editing to make something like designer dog breeds, which we already use artificial selection heavily within that? So definitely, I think we can all agree that to a certain extent, designer babies is like frowned upon and it's very concerning. But in areas we already use artificial selection so heavily, and the traits that we select for appear so quickly, is there really an ethical concern in that environment?
There are already gene banks, embryo banks, for people with Ivy League educations. You can actually choose a sperm donor or an embryo from somebody who went to the Ivy League, so it's already happening. So yes, traits are being sected out and chosen. And among, as you say, the well-to-do, you're going to see more people whose parents, whether they be the biological parents or surrogate parents, had Ivy League educations. And height, mention height. Well, people are taller now. One reason people are taller is tall people had a harder time surviving standards. And evolution selected them out. Now, the odds of anyone in America dying of famine, and especially anyone middle class or above, dying of famine is pretty minimal. And height's now been defined as attractive, and there's a genetic selection of people who are taller just for the sake of being taller.
diet also affects height. So
Yes.
poorer diet,
Yes.
which would be in times of famine,
Yes. Yes.
result in people who could have
Yes.
been taller being
Yes.
shorter.
Yeah.
Evolution, yeah, just cares who's having more babies. So the
Yeah.
genes for tallness are on this increase.
Yeah.
Fred.
Yeah, don't you think the genes kind of already have a bottle on this one? I mean, don't you think it's a matter of time before all the issue that an argument can be made that one gene can be changed and humanize stop colorblind and that becomes surgery that not only makes one person's world better, but it's also part of the game when they pass it on. I mean, like, usually when you gotta do play with, like, you know, I don't know, people play with it. I mean, whether ethically or not. Don't you think we're going to kind of run into problems with this, whether or not it's sanctioned use of the technology or it's unsanctioned?
Yeah.
That's what China seems to be doing. I don't know.
Amazon, CRISPR.
I mean, there might be some limits to the technology, so we might not be able to edit all 20,000 genes in a single embryo. That might be a useful limitation, I guess, in terms of this whole designer offspring kind of questions. But yeah, it's definitely out, and I think it's going to happen.
Dan, you had a question?
So I just had a question about in relation to like sort of like resources. So if you have somebody, let's say you have an embryo that we know has a genetic disease, and that could be cured, but we're like, oh, do we really want, who's to say if this is okay or not? Are there possibly some benefits that could be extracted from it, and this or that? But like there are known downsides, and perhaps these downsides later on in life, as it develops into adult, will lead to like treatment. Like you'll have to take like medication for it, and you'll have to pay for that, and then like if you can't afford it, maybe your insurance does, and then maybe they outsource some things, and then in the meantime, there's research devoted toward treating this disease that could have been more or less, or perhaps just more efficiently eradicated as a zygote or as an embryo when it's an embryo. So my question is, when it comes to, we could be devoting our time to less treatable things if we fix this problem earlier, and therefore devoting resources and research into less treatable things. Does it become an obligation to humanity as well as sort of the embryo as well? And where does that ratio lie to treat that or nip that in the bud in the embryonic stage?
I think if it's a genetic disease, if you treat the tissue in a human, then it still can be passed down to the offspring. So in some ways, germline embryonic alteration actually makes sense because you can remove that disease allele, at least one disease allele.
And sick people are expensive. Yeah. So, I mean, if you have a generation of sick people and then their children, yeah, financially and also medical and quality of life, of course, for the individual. Yeah. So sick people are expensive. If you can nip it in the bud, then perhaps you'll save all of those resources for something else, all of those doctors and research for sure. Think about it. And I think that the scary part about it is that gene editing actually makes a lot more sense when you're talking about it in germline. You know, on an adult human, there are very few diseases that you can fix with a gene that will, help their current problems.
But gene editing might actually be hitting it with a sledgehammer. Like, is gene editing necessary right now? Because this would be combined with in vitro IVF, right? So it'd be combined with in vitro fertilization. But in vitro fertilization is already using tools like pre-implantation genetic diagnosis to pick embryos that do not carry a disease allele. And you could just screen just a few genes that are known to be disease-causing. And so a couple that are carriers for cystic fibrosis or sickle cell could actually guarantee that their offspring won't get those.
Abortion rights are called pro-choice, but that may be a misnomer. Insurance companies require pregnant women to go through embryo to test whether or not the fetus has Down syndrome.